Chem test Actinomyces Neuii subspecies Neuii. In this video, we'll explore the history of the CAMP test and demonstrate the step-by-step procedure for performing this classic microbiological technique. We'll also discuss the unique interaction between Staphylococcus aureus and Actinomyces Neuii subspecies Neuii that leads to enhanced hemolysis. Don't forget to like and subscribe for more microbiology content. General information. Actinomyces Neuii subspecies Neuii is a bacterium that exhibits a unique reaction known as the CAMP test. This test, which stands for Christie, Atkins, and Munch-Peterson, is used to identify specific bacterial species based on their interaction with Staphylococcus aureus. To perform the CAMP test, a beta hemolysis producing strain of Staphylococcus aureus, ATCC 25923, is streaked onto a blood agar plate alongside Actinomyces Neuii subspecies Neuii. The two streaks are made parallel to each other, and the suspect culture is then streaked perpendicularly between them, without making contact. A positive CAMP test for Actinomyces Neuii subspecies Neuii is indicated by enhanced hemolysis in the vicinity of the Staphylococcus aureus streak. This synergistic effect is due to the interaction between the beta-hemolysant produced by Staphylococcus aureus and the extracellular CFP protein produced by Actinomyces Neuii subspecies Neuii. This phenomenon was first observed and described in 1944 by Christie, Atkins, and Munch-Peterson, who initially identified it in Streptococcus agalactiae. Dance of Lysis poem. A streak of gold, a streak of white, a meeting on the agar bright. Where Aureus meets Neuii's line, a crimson halo starts to shine. The CAMP test tells the tale of synergy that will not fail. A dance of proteins, lysis bold, a story in the agar told.